Isolation, Characterization, and Analysis of Streptomycin Mutations through the Medium of Interpretive Dance by Audrey Allison and Alina Stevens for Genetics Lab. First, E. coli was spread onto a TBAB plate with streptomycin. Then, a 10 to the 6th dilution was performed, and this was spread on 2 TBAB plates. All of the plates were then incubated overnight at 37 degrees Celsius. The next morning, the plates are removed and the TBAB plates are counted to determine the viable cell count. In our case, this was 1.325 times 10 to the 9 colony-forming units per milliliter. Then, the TBAB plus strep plate was removed from the incubator to determine how many mutants were formed. These mutants could either be strep-resistant, strep-dependent, or strep-intermediate. All of the mutants obtained have a mutation in the RPSL gene that codes for part of the 30S ribosomal subunit. Each of the mutants was then patched onto a TBAB plus strep plate and a TBAB plate. This was to determine which mutants were resistant, grown on both plates, or dependent and grew only on the strep plate. In our case, all of the mutants were streptomycin resistant, and the frequency of this was 3 times 10 to the negative 9th mutants per colony forming unit. There were no intermediate or dependent colonies formed. The E. coli was then suspended in water and added to PCR master mix, which included nucleotides, buffer, and Taq polymer. Then RPSL up primer and RPSL down primer were also added to the solution. And then the entire solution was placed into the thermocycler at 95 degrees Celsius for 10 minutes to denature the DNA, and then 30 cycles of 95 degrees Celsius to denature the DNA, 55 degrees Celsius to anneal the primers, and 72 degrees Celsius to extend the DNA, followed by a time at 72 degrees Celsius to finish all extension. The PCR process would exponentially duplicate the DNA to be studied. An MN PCR cleanup would be required to prepare the sample for sequencing. First, the sample will be loaded onto an MN column with NT buffer and then spun. At this point, the DNA would be on the column. The waste would be dumped and NT3 wash buffer would be added. Spinning again would occur, then water would be added, more spinning, and finally the DNA would be in the tube. A spectrophotomic approach was then used to ensure that the DNA had in fact been duplicated by PCR. The sample would then be sent off to GeneWiz, who would do the sequencing of the DNA. Several bioinformatic procedures were then done using the sequence. The first of these was BLAST-N, which compares the nucleotide sequence to the nucleotide database. The second of these procedures was BLAST-X, which translates the nucleotide sequence to an amino acid sequence and then compares that amino acid sequence to the protein database. Both of these procedures can be used to compare your sequence to the wild-type sequence to see what differences and similarities exist. Using these, we discovered that a mutation occurred at position 184, which was from A to C. This resulted in a change from lysine, that has a positively charged side chain, to threonine, that has a neutral side chain. The last bioinformatics procedure performed was multiline, which compared our sequence to the sequence of other students and the wild-type sequence. Thanks for watching!
Nothing on your side.